Hello. So, here's a question for you to start thinking about, and I'm fully expecting you to pause this video, do some deep reflection, maybe write some things down, and then come back to the video. So the question is, how are cells able to create the proteins that they or the body needs? So thinking about the process of protein synthesis, getting out a piece of paper, something to write with, and go. All right, I'm assuming that you just paused the video and that you have subsequently produced some magnificent diagram, maybe a long bulleted list of things that happen. And if you didn't, I want you to know that I'm disappointed. So here's what we're doing today. We are going to rethink the cell. And I mean that in the sense of we have a way that we've learned how things happen inside of a cell, and it makes sense, and we have good analogies about factories and post offices and things to help us to understand what organelles do. But I think it's valuable for us to deconstruct that and to reform it in a better way that fits more with the things that we've been talking about, and fits with this idea of the cell being this state of chemically controlled chaos. So first, here's a picture, nice picture of the cell, and this is better than a lot of pictures you see. It's got some three-dimensional nature to it. You can see that they've gone out of their way to show multiple mitochondria, right, because there's not just one. You can see the endoplasmic reticulum is this huge sprawling network all around the nucleus. It's not just one little blob. You can see the Golgi is kind of fit in amongst it. You can even see kind of sticking up out of the cutout here, finishing off sort of the tubes of part of the endoplasmic reticulum. So all in all, a pretty good diagram. Obviously, cells aren't all fancy colors and things like that. They're a little more blah, but this helps us to differentiate and also helps to clue us in on some certain parts that we typically see. You often see a nucleus with this sort of purplish color thanks to the dyes that we often use on prepared slides. And traditionally we see the orangish color for the mitochondria. It helps us to recognize these things. When we think about the process by which a protein is made, traditionally we think of it as a process that begins at the nucleus, because the nucleus is the thing that's in charge of the cell, the thing that's controlling what's happening inside of the cell. In fact, all of the organelles are these things that are working to control the cell. They're pushing the various buttons and pulling the various levers in order to make things happen inside of the cell. And so we think about how the nucleus makes RNA, a copy from its DNA. That's the process of transcription. And it sends that RNA out of the nucleus. That RNA finds the ribosome, and the ribosome then reads that RNA. The ribosome follows those directions in order to build a protein, so it uses the information from the RNA to pull together various amino acids in order to construct a protein. And then the ER might accept that ribosome with the protein that needs to be transported somewhere, right? That network of tubules that goes throughout the cell is ready and waiting for proteins to need to be shuttled off to some other part of the cell. And sometimes those proteins will end up at the Golgi, and the Golgi apparatus will package those proteins, might mark them for some sort of signaling that needs to happen inside or outside of the cell, and they might be sent inside the cell, they might be sent outside of the cell. It sort of depends on what the protein is and what the cell is and what the circumstance is, right? So I want you to think about those statements and try to think of, like, what are the problems sort of when we make statements like we just did. I wouldn't say that any of those are incorrect from a standpoint of understanding what these organelles are meant to do, but there is sort of a fundamental problem. And there's an assumption that's being made in each of those examples for the nucleus, the ribosome, the ER, and the Golgi. And there's questions that aren't really being addressed. So think for a second about those statements that we just made, which I think were fairly straightforward and pretty much by the book of how we've traditionally learned our organelles. So here's the problem. Organelles and proteins are not living things. And I know that you know that, right? If I asked you, is a ribosome alive? No. Is a nucleus alive? No, right? The cell is sort of the quintessential first characteristic of a living thing. You have to be on a cell level to even be considered alive. So we know organelles are not alive. And certainly we know proteins, which are just a macromolecule made of a bunch of amino acids, we know that they're not alive either. A second thing that I think that you know is that things that aren't alive can't make choices. They have no decisions to make, and they have no wants or desires or dreams for what they might accomplish in the world. Right. So we know these two things, and therefore, through the transitive property, something like that, we can put those two things together, and we can say, well, therefore, organelles and proteins, which are not living things, cannot make decisions. They can't intentionally do anything at all. And that starts to throw a wrench into the gears of how we think about a cell, because we talk about these organelles in ways that imply intent. And so let's look again at some statements. So the endoplasmic reticulum transports the newly created protein to its destination. Think about some problems there, or at least some questions that we should be asking in response to a statement like that. The Golgi apparatus concentrates, packages, and sorts proteins before they're sent to their cellular or extracellular destinations. Again, some problems. And the Golgi apparatus may add carbohydrates to the proteins. I mean, right away, the word may should set off some alarms. Like, it might? What decides if it does or doesn't? Certainly not the Golgi, because as we established, it's not alive. It doesn't make decisions. And so when we think about these statements, with each one, we can start to pick apart the things that it sort of glosses over, the decisions that are being made. What's the destination for the endoplasmic reticulum? There's a million different places you could go inside of a cell, so how does the ER know where to send these proteins? So these are the types of questions that we should be asking. So here's a better way of thinking about this. Stuff happens because chemistry. And that's how we need to be thinking about the cell. Chemical interactions are the things that drive cellular processes. They are the driving mechanism for what happens inside of a cell. The second thing we need to understand is that anytime we talk about something happening inside of a cell, we need to understand that it's not just like, oh, the RNA is going to the ribosome, which then goes to the ER. No, all of these things are happening many times in many places, all on top of each other. And the third thing to keep in mind is that diffusion is our friend. So let's think about this some more with ribosomes in the ER. So here's a fact. Sometimes, depending upon the protein that's being synthesized, the ribosome will attach itself to the ER because the ER is required to transport that protein to where it needs to go. So my follow-up to that is, how does this happen in a world where ribosomes don't know or want or hope or dream for things? So how does the ribosome know whether or not to land on the ER is the question. Either the chemical structure of the forming protein perhaps is chemically attracted to the ER in some way that draws it towards it. It's a reasonable guess. Or maybe, as the protein is forming, it causes a change or a shift in the shape of the ribosome itself. The ribosome is actually constructed of RNA and proteins itself, so it would be very susceptible to sort of chemical interactions around it. So that's also a possibility that the forming protein directs the change in shape of the ribosome itself, which then leads it to being more susceptible to an attraction with the ER. And I'll be honest with you, it doesn't matter which one of these is the answer. It doesn't matter. The point is that we understand that there's an underlying chemical interaction that's driving this decision. And it's not even so much of a decision in a traditional sense as more of a consequence of how it has to be because of these chemical interactions. So, if the ribosome or protein complex is attracted to the endoplasmic reticulum, it goes on over there, it cozies up, but how does it know where the ER is? And that's the next question. So think about that. How does the ribosome know where the ER is to even sort of have this chemical attraction or interaction? Because it's not like you could have the ribosome on one side of the cell and the ER on the other side of the cell, and it's like a tractor beam pulling them together. That doesn't make sense, right? The cell is, on that scale, too big, and the chemical interactions we're talking about are like hydrogen bonding and things, the weakest of attraction. So it's not like the ribosome just goes flying off through the cell with some sort of homing beacon for the ER. So how does it know where the ER is? It's a good question. It doesn't. Ribosomes don't know things. And frankly, I'm disappointed that you weren't listening well enough to immediately answer that question. Ribosomes don't know things. It doesn't know where the ER is. It has no idea. So how does it get there? Remember, everything's crazy on a chemical level. The cell is a chaotic mess of chemistry. Stuff is in constant motion. Stuff is in constant flux. And really, it comes down to, believe it or not, chance. The ribosome, as it's zipping around and bumping into things, is eventually going to find that spot that it fits into, and eventually will let that protein go, and those chemical interactions win out, and the desired conclusion is reached. And I do realize I said desired outcome. Again, ribosomes don't actually care whether or not they find the ER. In fact, you could imagine a poor little ribosome who just by chance, he's got the protein ready. He's ready to drop it off, but just by chance, he never finds the ER. It's very sad. I think Pixar needs to make that film because I would watch it. So let's recap. Stuff happens because of chemistry. Chemical interactions are the thing that drives cellular processes. Second thing, everything we discuss about cells is actually happening many times in many places within the cell, and diffusion is our friend. So we have to remember these statements as being sort of the underlying reason behind what's going on. We can think about a ribosome potentially having trouble finding the ER because let's say that ribosome was making insulin. You just ate one of those 3-foot-long pixie sticks, and your body needs insulin, or your heart's going to explode from the amount of glucose that's suddenly entering your bloodstream, right? So you need that insulin immediately. Not like, we got like 30 minutes and we'll slowly build up insulin and hopefully those ribosomes find the ER. That's not how it works. These things happen very, very quickly. Our body processes are they're not immediate, but they're very close. So when we think about how is it that the ribosome finds the ER, again, it doesn't find anything except through chance. It's not deliberately going out and seeking the ER. Instead, what's happening is it's randomly moving throughout the cell and it bumps into the ER and eventually delivers the protein appropriately. But think about that second point on the screen. There's not just one ribosome. We didn't just make one copy of the RNA. In fact, starting in the nucleus, we made tons of copies of the RNA programming for additional insulin. We delivered those copies of RNA to tons of ribosomes through chance interactions again, and those ribosomes are delivering those insulin that they're producing to the ER and to the Golgi and subsequently out of the cell at a very, very rapid rate and at a very, very high number, which is why we can count on it happening. Yes, is there that one poor, lonely ribosome that just can't find the ER? Probably, but that's okay because there's a few hundred or a few thousand that are managing to do it in that same cell or in surrounding cells in countless places throughout the body, and that's able to accomplish the goal. So the chaotic nature of the cell on an individual interaction level seems problematic. But we have to start thinking about this on a more large scale and understanding that the cell is not just doing this one thing and it's following this process and the ribosome is making the protein. That's never how it's working. Instead, we have countless ribosomes making tons of different proteins, and many of those ribosomes might be making some of the same proteins, and the shifting amount of how much of this protein versus that protein and all of that is changing because of signaling inside of the cell, controlling and maintaining homeostasis, but all of it is governed by chemistry. So when we talk about this stuff and we talk about why does this happen, why does even the mitochondria accept the broken down products of glycolysis in order to undergo the Krebs cycle and the ETC? Why does this happen? Chemistry. Chemical interactions, chemical attractions, diffusion pushing things into different places, active transport creating circumstances where things get pumped and moved across membranes in a variety of ways. But at the end of the day, every single one of those things comes down to a chemical interaction and a chemical attraction, because stuff happens because of chemistry. I hope this was helpful, and we'll talk about it more soon.